Good morning, friends. In the series of ultraviolet visual spectroscopy, we have learned many topics, theories connected with ultraviolet visual spectroscopy. In the last lecture, we have learned about colorimeter and basic instrumentation involved in the colorimeter, theories involved in colorimeter. Now here, any instrumentation involving spectrophotometer or either colorimeter. Now the block diagram is similar. Here one light source is there, here slit will be there, here filter will be there, here cuvette will be there, detector and recorder system will be there. That means this should be the block diagram of any spectrophotometer or colorimeter or any photometer. So basic difference is nothing but now we will see one by one what should be the major involvement or major steps one by one. Now here filter. Now we will go through this deep study of filter. Now filter acts as a wavelength selector. Here filter acts as a wavelength selector. Now if we see here, wavelength selection takes place either by prism or diffraction grating. Now we will see in this lecture, this lecture will focus on the working of prism in wavelength selection system. Now in any technique of colorimeter and spectrophotometer instrumentation, it is similar. The instrumentation is similar. Colorimeter and spectrometer instrumentation is very similar, but the mode of measurement and type of detector are different. The measurement system, in which way it deal with, and the detector, the different detectors are employed to detect the intensity of transmitted radiation, that should be different. The color of the filter must be therefore either complementary color of the solution. The pair of the colors which when mixed with give the effect of white light is called as a complementary color. Now the complementary color, the transmitted light that is gives the interesting topic, that is a complementary color. The color of the filter must therefore be the complementary color of the solution. Now the we know that every colorimeter or spectrophotometer that deals with the colored substance. Now if the colored substance is not should be if the analyte is not colored, then that analyte should be converted into the colored form by using some suitable complexing agent. Now the color of the filter must therefore be the complementary color of the solution. Now the concentration of the color that will decide the intensity, and from that intensity of the color, concentration of the analyte or concentration of the solution that can be measured with the help of colorimeter or spectrophotometer. The pair of the color which when mixed to give the effect of white light is called as complementary colors. So here complementary color, if you know sun, if you see the rainbow, then the rainbow is nothing but it is a sense of complementary color. If we take the example, a white light is mixture of different colors. So in that different colors from different shades are available. So that is all about the complementary colors. A narrow band of wavelength is always to be used in absorption measurement. Now for this purpose, filters are simplest and least expensive. So filters aren't why filters are used? Because they are the simplest in working, and they have the cheaper or least expensive. This was the a narrow band of wavelength is always to be used in absorption measurement. So in every absorption measurement, we do not use the broad absorption of wavelength. We use the narrow or selected beam of radiation of wavelength. The fourth paragraph, filters are used in colorimeter for producing limited band of radiation. Now filters are employed to produce a limited band of radiation. Filter is simply a physical object which when absorb the light of some wavelength wholly or partially and allows the light of required wavelength to pass through it, that means transmitted radiation. There are mainly two types of the filter. One is absorption filter, another is interference filter. So that is all about the filter system. Now filters are nothing but the wavelength selectors. Now some of the instrumentation we use the prism which acts as a filter. So prism is we can say it is a unique one. So prism is nothing but the it is a triangular shaped geometry. A prism has a triangular shaped geometry. Now we will focus on the here prism and its working. A better prism, in that prism we can say better optical system is needful to improve the resolution of spectrum in the both visible and ultraviolet region. Now here better optical system is needful to carry out or to measure the readings related to colorimeter and we can say ultraviolet UV visible spectrophotometer. This is achieved by use of prism. Now the prism is nothing but the, you can say, either here prism, the diagram of prism is given. This is all about the you can say basic principle and working associated with the prism. So this is achieved by the use of prism. The action of prism depends on the refraction of light. This is the most important. Refraction of light by the prism of materials. So this is a basic principle of prism. So that prism acts as a acts on the refraction of light. Now there are mainly two types of the prism. One is glass prism, which plays a role in visible region. Another is a quartz prism. Quartz, it is made up of quartz. The material is made up from quartz. So quartz prism are employed in ultraviolet region. So two types of the prisms are used in the absorption measurements, mainly glass prism and quartz prism. Glass prism is useful in visible region, while quartz prism is useful in ultraviolet region. When a polychromatic light is filtered through a prism, dispersion of radiation takes place according to wavelength to different wavelength. Now the prism acts on a refraction of light, but when a white light or polychromatic light is allowed to pass on the surface of prism, at that time dispersion of radiation or dispersion of light takes place to a particular wavelength at a different wavelength. So we get a continuous spectrum of the wavelength. So continuous spectrum of radiation is obtained. Now exit slit. Now we use to avoid this interference, we use the exit slit. What is the role of exit slit? Exit slit is used to select the wavelength. Proper wavelength or suitable wavelength is obtained from the exit slit. Prism is rotated slowly so that the radiation of particular wavelength is allowed to pass through slit. So here prism is rotated in such a way that we get a proper wavelength, we get a suitable wavelength. Now what is the advantages? What is the advantage of prism? Prism produces a single order spectrum. A single order spectrum, it is the most important advantage of prism. A single order spectrum is obtained from the prism. What is the disadvantage of prism? It gives the overlap of wavelength. Overlap of wavelength, that is the main interference associated with the prism. So this is the disadvantage. Now if we see, this is a diagram of prism, diagrammatic representation of prism, which are placed in the any absorption measurement like colorimeter or ultraviolet or ultraviolet spectrophotometer, ultraviolet visible spectrophotometer. So here prism acts as a filter. So white light is passed and entrance slit is allowed through a entrance slit. White light is passed through a entrance slit. Now this white light, this black line indicates you can say the mode of white light, way of white light bombarded on the surface of prism. Now here you can say dispersion takes place and here refraction takes place. Here refraction takes place. Now this refraction takes place into different 7 colors. Among that, this is the entrance slit where this light come out. Violet bend. Now here violet color. This is a violet color. This violet color mostly it bends. Bending of violet color takes place at a large extent, while bending of red color takes place to a lesser extent, to a least extent. So that is the main difference we can say between the violet and red color. Now the red color is obtained in the visible spectrum of electromagnetic radiations, while violet color is obtained nearly UV region of the electromagnetic radiation. So we have the different complementary color also. Now we use the if the solution is purple color, then we use the green color filter for the measurement. If the bluish green color, or can say if the red color is the solution has a red color, then we use the bluish green filter for the reading purpose. So on the complementary color, on the basis of complementary color, we modify the filter, we modify the prism. Okay, so we modify the filter also, not a prism, because prism is, you can say, either it is made up of either glass material or quartz material. So this is all about the basic thing about the principle of filter. What is the principle of filter, why it is used, what are the cracking difference between the filter and prism. So that is the most important. This lecture will give the idea about all this. With this content, I conclude this lecture. Thank you very much. Thanks for patience listening.